This is Module 3, Oxygen Binding Proteins and Methods for Analyzing Proteins. In this video, Common Laboratory Techniques for Protein Analysis, I'll be describing several different methods for how we quantify proteins in the lab and also characterize proteins in the lab. So for the rest of this module, I'll be discussing protein analysis methods. First, I'm going to talk about easy ways to quantify the amount of proteins you have in solution in the lab. This is done through absorbance spectroscopy, which measures the total protein concentration in a solution. This is actually dependent on the relationship between absorbance and transmittance. So if you'll think back to when you first learned about this, about spectroscopy, also called spectrophotometry, that's why I keep messing up that word. You have a sample cuvette of proteins in aqueous solution, and you shine a light on that. The amount of light that is transmitted through to the detector is a measurement or inversely proportional to the amount of light absorbed by the sample. And so we can measure the amount of light absorbed by the sample, and that will give us an indirect measurement of how much protein we have in that sample. The proteins in a sample can only absorb light if they have one of these three aromatic compounds. And nearly all proteins will have some varying degrees of these aromatic compounds. And so this still works. Histidine absorbs light because of its aromatic R group. Tyrosine absorbs light for the same reason, and tryptophan absorbs light at the highest amount because it has two of these aromatic rings. And so UV light can be absorbed by these aromatic rings. So you have to use the right wavelength of light, UV light, and so that's going to be somewhere here in the center. Protein molecules that are high in tryptophan will do the best, and so it will be easier for you to quantify those proteins versus proteins high in histidine, which don't absorb as much light. But again, most proteins contain mixtures of all three of these, and so this is one way you can indirectly measure how much protein you have dissolved in aqueous solution using light absorption techniques. Okay, so let's talk about how we study individual proteins in the lab. If you want to study a cellular protein, you have to find a way to separate it from all of the other cellular proteins. And this can be fairly tricky. But if you know enough about the properties of your protein, you might be able to winnow down your sample from all of the possible proteins that could be present in a cell to maybe a handful of proteins which contains your protein of interest. And so the first thing you do is you create what's called a crude extract, where you break cells open in a lysis buffer, but you don't take anything out. So you take those cells, drop it in a lysis buffer, which tends to be like a soapy lysis buffer. That breaks open the cells, and all of the proteins in the cell spill out into the surrounding aqueous environment. This means your protein of interest is actually mingling with thousands of other proteins in the cell. And so you want to be able to progressively lower the amount of contaminating proteins, which will increase the concentration of your protein of interest. So the first step is to precipitate proteins with similar biochemical properties to your protein of interest. The second step will be to use specific purification methods depending on that will isolate out your desired protein based on differences in size, charge, or specific interactions with molecules if it is a protein that binds a ligand. So let's take a look at this first step first, how we precipitate proteins with similar biochemical characteristics. We start with a crude extract, and it's typically put through one first procedure called salting out, where you increase the amount of salt concentration in your solution to the point where other dissolved compounds in solution fall out as a precipitate. Remember that water molecules are very attracted by salt ions, and that decreases the number of water molecules available to interact with a charged protein that's in solution. Protein molecules, if protein molecules lose the ability to interact with water, they will start to coagulate by clustering together with each other. And when that happens, this is called salting out. So here's our salt concentration on this axis, and this is the solubility of the protein. When salt concentration is low, here's your protein here, And then you have these water molecules interacting with this protein dissolving in the solution. If this is a globular protein, there's going to be polar residues on the outside of that protein molecule interacting favorably with water. As you increase the amount of salt in that this system, So as we increase the amount of salt, again, you're going to have water molecules forming solvation shells around your protein molecules, and that will break up and dissolve any clustered proteins that are clustering together. So if the proteins are clustered together, initially adding a little bit of salt actually encourages them to dissolve a little bit better in water. But if you increase the salt to an insane amount, like a ridiculous amount of salt, very, very high molarity, then the proteins will cluster together because the water molecules are too busy dissolving the tremendous amount of salts in the solution. Different proteins salt out at different salt concentrations. So you can start sort of toying with pulling out your protein of interest by looking up, methods in the past that have worked for salting out particular types of proteins. Okay, so now we have a precipitate, and this still consists of, so this is showing a very simple situation where your target protein is the only one that is salted out, but that's not very likely. It's probably going to salt out with other similarly charged or sized proteins that have similar biochemical characteristics. And so you have to continue to remove the contaminating proteins in order to examine your protein of interest. One way to do this is through a process called column chromatography, which separates proteins based on their specific biochemical properties. We start up here where you put your protein sample at the very top of what's called a stationary phase. This is a column that has some type of solid porous matrix, and the nature of this matrix differs depending on your target protein. So I'll talk about that in the next few slides. Up at the top is a reservoir of water that's dripping down onto your protein sample, and this forces the protein to move through the solid porous matrix. What you get at the bottom is proteins will move through at different rates depending on their biochemical properties. And so when you open up this channel at the bottom, first you're going to get some waste to liquid coming out from the column, and then you're going to get proteins separating out one at a time based on their biochemical characteristics. So let's look at how we can do this based on different sizes of molecules, different size proteins, different charges, or if they are binding proteins that bind to specific ligands, we can use that characteristics to purify them as well. Let's first look at chromatography methods that separate based on size. So this is a column chromatography method called size exclusion chromatography, otherwise known as gel filtration chromatography, which separates proteins based on size. In the solid porous matrix of this column, there are beads that have pores in them. The most common type of bead is to use a silica bead or silica gel. As proteins move through this column, very small molecules, so small proteins, will kind of fit into these pores within the silica beads and have a very difficult time migrating quickly through that matrix. Very large proteins won't get stuck up in these pores and will move through at a quicker rate. And so when you open up the elution at the bottom, very large proteins will come out first, followed by the next smaller proteins, and they'll come out in order of size. And so if you are trying to purify a protein that you know to be very large, what you would do is put your total protein mix. First, you would do a crude extract, then you would do your salting out, and then you would put the total proteins up at the top. And if you're looking for that large protein, you would elute for a certain amount of time that only allows large proteins to exit the column, and small proteins will remain in the column. So again, small molecules kind of get stuck up and take a longer path through the porous particles of the solid matrix. Larger molecules kind of move right through it, so they come out first. Let's look at a column chromatography method that separates proteins based on charge. So Remind yourself that pH, the pH of the surrounding environment, will affect the charge of an amino acid. And so what you first have to do is find the ionizable groups, look up the pKa of each ionizable group for each residue, and determine if it's protonated or deprotonated at a particular pH. If pH is lower than the pKa of that group, then it will hang on to its protein. If pH is higher than the pKa of that ionizable group, then it will lose its proton. I think I said protein, I meant proton. So it's protonated if pH is lower than pKa, it's deprotonated if pH is higher than pKa. This will allow you to determine the charge of each ionizable group in a chain of polypeptides. Usually you don't have to start out doing this if there's something already known about the protein. This information is available on databases online. So this has already, to some extent, been worked out for you. This is just an example of how you can do this. So if your ionizable group is the carboxyl group and it's in its protonated form, because pH is very low, then the charge of that group is 0. And let's say we stick to that low pH, that means that the amino group is also protonated, which has a +1 charge. And so the overall charge of this would be +1. If you increase the pH to the point where you're very, very basic, then this carboxyl group will lose its proton and become negatively charged. This amino group will lose its proton and become neutral. And so pH really does affect the charge of the amino acids and will affect how something moves through a charged stationary phase in a column. The column chromatography method that separates proteins based on charge is ion exchange chromatography. In the stationary phase of the column, we have either positively charged beads or negatively charged beads. The positively charged beads tend to be these beads called DEAE, and they are going to bind to and interact with negatively charged proteins moving through the column. The negatively charged beads are going to bind to and interact with positively charged proteins moving through the column. And so when you have anion exchange or positively charged beads in your stationary phase, you add your proteins to the top and you allow them to move through. That's going to actually mean that your your negatively charged proteins will get trapped in the matrix, and then the other ones will move out. And so you can open up the solution at the bottom, allow everything to move out, because the negatively charged protein of interest is going to be bound to the positively charged beads in the column. Once you get rid of all of the things you don't want, add a high concentration of salt that will disrupt the ionic interactions between the positively charged beads and the negatively charged protons. So similar to the idea of salting something out, and that will cause your protein of interest to come out at the elution on the bottom. So to review, in anion exchange chromatography, negative proteins will stick to the positive charge of the DEAE bead. Positive proteins and neutral proteins will flow through and be discarded as waste. Then you add your salt, your high salt, which will, or you could also alter the pH of this environment, which will disrupt ionic interactions between the positively charged beads and the negatively charged proteins, causing the negatively charged proteins to move through the column and out on the bottom. So there's also cation exchange chromatography, which is the opposite case, using negatively charged beads to trap positively charged proteins. Now let's look at column chromatography methods that separate proteins based on what they interact with. Remember that some proteins are actually capable of binding ligands. So enzymes bind substrates. Receptors can bind hormones or growth factors. Antibody proteins bind antigens, pathogenic antigens. Transcription factors, which are proteins that help to encourage gene expression, will bind to DNA as its ligand. As we reviewed in the first 2 videos, hemoglobin and myoglobin bind to oxygen as their ligand. And so what we can do is design the stationary phase of the column to be covalently attached to the ligand that specifically binds to your protein of interest. So if your protein of interest is this half moon, your ligand is the missing piece that binds to it. So here we have your mixture of proteins. Some of them are half moons, your protein of interest. Some of them are these triangles, some of them are these squares. And so the triangles and squares are not going to bind to the ligand that's covalently attached to the bead in the column. And so those will move through on a short path and come out first. The protein of interest are going to bind to the ligand on the column. And so what we can do is, so those get trapped in the column, get rid of all your waste first, get rid of all those unwanted proteins. And then remember in the last example, you add a bunch of salt to disrupt interactions between the bead and the protein. Here you would add a high concentration of ligand, which is going to encourage the protein to come off of the bead and maybe bind to the ligand in solution. These are reversible binding events, so this happens to various degrees. And so if you add high concentrations of ligand to this, then the proteins will bind to those and come off at the end of the column in the elution stage. You can also use high salt to disrupt the interaction between the ligand and the protein of interest, because that interaction is via non-covalent reversible bonding. So if you add a lot of salt, that disrupts those binding interactions, and the protein of interest will elute out at the bottom. Okay, so what we've been essentially doing this whole time between creating a crude extract, which is not itself a purification step, but it's just preparing your sample, and then doing your salting out procedure, and then any chromatography procedures, what you start with is this high mixture of proteins where your protein of interest is the red spheres. What you end with after all of these purification steps is a higher concentration of your protein of interest compared to the contaminating proteins that remain. We can measure this so we can measure the amount of protein of interest after the purification steps using the term specific activity. This is only possible, though, if your protein of interest has some kind of action. So this is really only useful if your protein of interest is an enzyme and has the ability to turn a substrate into a product. The specific activity is the number of enzyme units per milligram of total protein. And so the specific activity in this beaker is very low because the number of enzyme units here is 5, and there's lots of contaminating total proteins. Here, the specific activity is much higher. Still, we have 5 red spheres, so the same number of enzyme present, same amount of enzyme present, but fewer contaminating total proteins. This means specific activity will increase with each purification step and will become maximal and constant when the enzyme is pure. And so activity is measured as this enzyme's ability to turn substrate into product. Okay, what if you want to detect the presence or absence of a protein in a sample? So you're not necessarily interested in separating it away from all of the contaminating proteins, but you just want to know if your sample contains a protein or not. This is something that's common if you're trying to understand or diagnose cancer, for example. And so cancer cells express proteins at different times, times when they shouldn't be expressing those proteins, which disrupts the ability for the cell to control its division rate. If you want to know if somebody's tumor has this cancer protein in it, then you don't need to do all these purification steps and isolate that protein away from the rest of the cells. All you need to do is find a way to light up the cancer protein in a way that you can see it compared to the rest of the proteins in the sample. And so it's a presence-absence test. One way to do this is by using SDS gels or SDS-PAGE. SDS-PAGE is an acronym, and it's this crazy acronym. Sodium dodecyl sulfate polyacrylamide gel electrophoresis is that, but I am never going to call it that. I will always call it SDS-PAGE. This name refers to the different steps involved in running the procedure. So SDS is a detergent that actually denatures proteins. So the first thing you have to do in preparing your sample is to add mercaptoethanol. Remember this reagent? This came up during the Anfinsen experiment in video 2-4 when we talked about the chemistry of protein folding and how Anfinsen proved that denatured proteins will spontaneously refold in aqueous solution. So review that video if you can't remember what this is used for. Mercaptoethanol specifically breaks disulfide bonds. So it breaks the disulfide bridges that are formed between cysteine residues on a polypeptide chain. Once that covalent interaction is broken, we then have to disrupt all the other non-covalent interactions holding this protein together so that it migrates specifically based on the length of its primary structure. So SDS will bind to proteins and cause the protein to unfold, and also will give it an overall negative charge. Very small molecules have fewer negatively charged SDS molecules bound to them and will move more quickly through the gel. Very large molecules have more SDS molecules bound to them and are very cumbersome, big, fat molecules that migrate very slowly through this acrylamide gel. And so the first step is disrupt your disulfide bridges. So I just reordered these steps a little bit to make more sense. So first we add mercaptoethanol to break the only covalent bond that holds together a tertiary structure, the disulfide bridge, between cysteine residues. We then add SDS, which will disrupt the non-covalent bonds holding that protein together, so it will unfold. It will also coat everything with a negative charge and separate proteins based solely on its size, not on its charge. We then cast what's called a polyacrylamide gel, which creates like a porous matrix for the proteins to move through, similar to the type of solid matrix in a column. And so negatively charged protons will move towards the positive electrode when an electric field is applied to this gel. And again, very large molecules will get caught up in the gel, stuck up there as they're very big and cumbersome and won't have the ability to easily navigate this matrix. And very small molecules will move more quickly through the gel. You then can take that gel and apply a membrane to it. So you just take a nylon membrane and press it onto the gel. And those proteins will migrate from the gel onto the nylon membrane. So that, you don't always have to do this. Sometimes you can just look at the gel itself, the SDS-PAGE gel itself. And this is what it looks like. So the proteins will migrate through the gel based on molecular mass. So based on the length in mass of its primary structure, the order of amino acids linked by peptide bonds. If you want to transfer this these gel results to a membrane, you have to add another step called Western blotting. And so it's not enough just to run the gel. You can't really do too much with a porous gel. They're very delicate and they're also very toxic. So you need to find a way to move the proteins, the separated proteins that look like this, out of the gel and onto a more solid substrate that you can manipulate and move around. This solid substrate is a nitrocellulose membrane, which is positively charged. Remember that all the proteins that moved through your gel are negatively charged. And so what we can do is create a situation where we're encouraging the negatively charged proteins in the gel to migrate to the positively charged nitrocellulose membrane. So we take the gel, put it on top of the membrane, put it in between a sandwich of filter paper, just as an easy way to separate it from the cathode and the anode. This is called a sandwich for obvious reasons. It looks like a sandwich. We have a negatively charged cathode here and a positively charged anode here. And so when the electric current is applied, the negatively charged proteins will move towards the positively charged anode and get trapped on this positively charged nitrocellulose membrane. And you can pull that membrane off. And what's really cool, and I don't have a picture of it, but what's really cool is the membrane will look exactly like your gel. You can't see it though, it's not visible. And so what we have to do is make your protein of interest visible for detection. So now we have your membrane, which contains your target protein, and we can visualize your target protein using antibodies that bind to it very specifically. Antibodies bind their protein target with high specificity. And so if we design an antibody that binds your target protein, it will only bind this target and none of the other contaminating proteins in the gel. So the first step is to design this primary antibody that binds your target and apply that to the membrane. These are going to be completely different for every target, and so it's difficult to visualize this because you'd have to come up with a different system for every single antibody ever made, which is just too many. And so what we do is we create a secondary antibody that's capable of binding every type of primary antibody. The primary antibodies bind to their target proteins with specificity only on the forked end, but this long shaft end is the same for every single primary antibody. That binds a secondary antibody, and that secondary antibody is conjugated to a colorimetric substrate, some type of protein that when cleaved by its enzyme will result in a color. And so what we are seeing when we see this color is an indirect measurement of how much target protein is actually stuck to your membrane, and that protein is stuck to the membrane by transfer from an original gel.